The innovation in cancer research and cancer drug discovery that we've experienced over the last decade, by anyone's account, has been truly remarkable, really fueled by a number of things. Certainly a much deeper knowledge in cancer mechanisms, the application of human cancer genomics, and the study of new approaches. There are many, but approaches such as epigenetics, tumor metabolism, and what you've heard a lot about recently, anti-tumor immunity or cancer immunotherapy. So as a result, cancer pipelines are currently quite rich in diversity, but they still face the challenge of really defining the right patient population for novel drugs and novel drug combinations, and also bringing these drugs to the market in a cost-efficient and a timely manner. So to discuss these and other issues of cancer drug pipeline development, we have today a group of leading experts, both from academia and the biopharma industry, that I'd now like to introduce. To my left is Jay Bradner, president of the Novartis Institutes for Biomedical Research, George Demetri, director of the Center for Sarcoma and Bone Oncology and senior vice president for experimental therapeutics at the Dana-Farber Cancer Institute, Steve Farwell, vice president and head of oncology, eye science at AstraZeneca, Keith Flaherty, director of clinical research at the Mass General Hospital Cancer Center, and finally Sean Harper, executive vice president of research and development at Amgen. Welcome. So I've taken the liberty of assigning you guys a few questions, so I'll just start out that way, but feel free to chime in to any question you'd like to answer. And I think we're also supposed to have some questions from the audience if I could figure out how to use this iPad and read the print. So maybe I'll start with the large font over here after I get my glasses on. Okay, maybe starting with you, Sean. So oncology has really been a very active, arguably perhaps the most active therapeutic area for drug development for many pharmaceutical companies in the last several years. So first, do we see this trend continuing in the pharmaceutical industry? And maybe venture your thoughts on why oncology has been so highly prioritized in pharma pipelines, other than the obvious, which is the clear unmet need.
Yeah, I would imagine that we're going to continue to see a very significant emphasis in oncology from the biopharmaceutical industry. And I think, as you mentioned, what motivates many of us who've joined the industry from academia is the fundamental unmet need. And certainly at an emotional level, I think there's no disease state that resonates with people more from an unmet need perspective than cancer. So I think that drives a lot of investor interest and the drive for people in the industry to want to respond to that. None of us are fortunate enough to not have close family members or friends who've been afflicted with this disease. I think also the field has moved into this critical zone where enough is understood now, predicated on the last 15 years or so of molecular biology, the advent of high throughput sequencing, the ability to demonstrate in multiple times that engineering-based approaches to particular targets can be successful. And most recently, the immuno-oncology kind of revolution, which I think is particularly exciting to those of us who have thought about this problem deeply and consider it to be very similar to the challenges one faces trying to eradicate a complex infectious disease such as tuberculosis. And so I think we all understand that it's going to be complicated, it's going to require combination therapy, but the fact that you might be able to engage the immune system to mop up and get you to a final, we had a little debate in the prior panel about what cure means, but to get to cure is starting to feel like something that you can get to in not as an exception, but perhaps as the rule, although that won't happen overnight. So I do see it. We're at Amgen very diversified, and we don't focus the company entirely in oncology, but I also see it as being part of our core mission to pursue it, and I think the time is ripe.
Steve, do you want to comment on that?
Yeah, no, it would be to highlight many of the same points. I think it's a disease where our molecular understanding of that disease has really evolved over the last 10 or 15 years. I think we've seen tremendous successes with targeted therapies and the ability to select patients, populations who are going to best respond to those therapies. We're beginning to focus on resistance mechanisms that emerge and come up with next generation drugs or a combination of drugs to try and deal with that challenge. And as Sean said, obviously the revolution of immuno-oncology has offered up that hope of potential cures, or if not cure, the idea of taking people and giving them years of life rather than simply weeks or months. And obviously that should be all of our aspirations. I think it's been this confluence of the technologies as well as a few new areas of biology that have really exploded.
Great. I'm going to ask Keith and or George a question that wasn't part of our prepared questions. You've both interacted a lot with the pharmaceutical industry. Do you feel that your interaction, your influence on the pipelines, the approaches that are being developed are being heard, and how might you like to see things perhaps a little bit differently? George?
May I start?
Yeah,
so
please.
I actually think that Keith and I have very similar experiences that are very positive about taking an academic approach and then bringing it to pharma and saying, we can't do this without you. This is a partnership between us and academia, between our patients, and between the pharmaceutical industry, be it biotech or big pharma. And I think there's all ears on that problem right now, because just what you've just heard from our colleagues at Amgen and AstraZeneca is the idea that we've got technology now. We need to now harness and focus that technology. So if we have an idea, if we have an insight in academia, I've found nothing but open doors and open ears in our colleagues in the pharmaceutical industry to make a difference for patients. So Keith, I don't know if that's your experience as well.
Yeah, no, I think it's right. I mean, it's whether it's large pharma or small biotechs, which is I don't know, any given day the number varies, but I'd say maybe 50-50, a split of where agents are coming to us from in the sector. I'd say basically the conversation's similar. It's just that the sense of panic in small biotech, which is X number of months/years and $25 million in the bank to prove a few points. Creates a heightened sense that you got to demonstrate, at least proof of mechanism, if not proof of concept, along the way. But big pharma wants to do the same, right? I mean, Jay's managing this, preposterous portfolio of agents, and some agents can't go forward. I mean, they have to do their molecular job, and ideally some version of a clinical job. So I think that there's harmony in terms of the idea that one has a scientific hypothesis, the drug's supposed to kind of ultimately contribute to proving that hypothesis. And I think the field has aligned very nicely along that early drug development mission as a way of then deciding what to prioritize and what not. I think a point to raise and maybe circle back to is the bigger challenge, which is that what Steve's alluding to ultimately, in my mind, is kind of the Weinberg and Hanahan problem, which is that we've got this whole wheel of cancer biology components. Some are better fleshed out than others. So we've got some oncogene targets. Okay, check the box. We've got a handful of those. We have some tumors that are susceptible to single drug immune interventions, but just some. And even then, there's such a thing as acquired resistance. And we're still just getting warmed up in epigenetics, metabolism, and a bunch of other areas. And ultimately, if we're going to get to the HIV-like model of, if not cure, then long-term survival with this disease, which is still kind of where I think I'll be retiring, in that era, then we need to understand the impact of each drug in the other components of biology, right? Which is we need to understand the immune consequences of targeted therapy, the effect on tumor cell autonomous biology with immune treatments, what subclones are picked off, which ones are not. And I don't see the field yet grappling with that problem, right? You're going to develop a given drug and you're going to establish that it does its molecular job, and maybe it can combine with standard of care therapy and make it to market. But if we don't understand how it actually fits into the bigger puzzle, then I think we're really challenged. And of course, I don't really task the $25 million Series A funded company with solving that problem. And Novartis and Amgen and AZ shouldn't be tasked with doing it each themselves. But we do need to come to a day where we see this as part of our shared enterprise. And I say the same thing to everybody. I mean, not just today's discussion, but beyond. We just have to come to a realization that it's not going to be a matter of just throwing drugs over the fence of FDA approval, getting them in the market, but actually figuring out what each brings.
Great. Okay, let's talk a little bit about the types of cancer and how we're going to approach them in the future, specifically, again, talking about the biopharmaceutical industry. So do we see things being discovered and drugs being discovered and developed along specific cancers, or do we think about a different approach in the future? And as part of that, how do we think about the role of molecular stratification as we develop drugs in the future? Keith, maybe I'll continue with you and then ask George and perhaps Jay to chime in.
Yeah, I mean, I guess I'm a big advocate of the imatinib model, carried forward, which is still to seek the opportunity for establishing proof of mechanism and proof of concept quickly. And that might mean doing it in a rare population where the cancer biology is wired in such a way that there's a real vulnerability. To be able to establish, okay, well, at least we can show here that we've got a real drug. It gets around, gets to the relevant compartments and has its effect. That might be 1% of the real cancer population for which that therapy is going to be relevant in the future. But I think there's a notion that there's some low-hanging fruit for given therapies that support the idea of picking off either tumor by site of origin or probably more relevant is the molecularly defined subset within given a certain set of cancer populations, which might still boil down to rare populations. So staggering development that way, I think, is prudent for everybody, whether big and small in this space. Again, it still doesn't resolve the problem of trying to figure out how it fits in, right? So it may well be you can produce high response rates and durable response rates in this really kind of simple context, if you will. I mean, a genetically simple, let's say, tumor type. And yet when you go big game hunting for the rest of the population you want to treat, there's compensatory mechanisms and clonal heterogeneity that just make that tumor look like a really simple, almost benign problem. And you may learn relatively little about how to tackle that. But I do think there's an element there to kind of de-risking and graduating therapies in a way that's still very, very useful. I mean, imatinib maybe is the right and wrong example because it found a home and kept a home in a couple of tumor types where the wiring was such that it's a really intricate link, and it's not as though we use imatinib to treat breast, colon, prostate, and lung. We haven't figured out that nut. But for many therapies, and I think with the immune therapies as well, in melanoma, my little corner of the world in terms of tumor type of greatest interest, lots of investigators and pharma sponsors as well look at melanoma and say, well, that's a feeding frenzy that's already kind of picked off. But is it still a population where there's a powerful proof of concept work that can be done? Yeah, I'd say until we learn more about how to figure out how to move the needle in pancreatic cancer in the same way, that you could still make that point. And I don't mean that to suggest that we're kind of using our patients to figure that out. It's the opposite. It's using that context to learn, innovate, serve the population along the way to figuring out how to actually move those therapies to other tougher nuts to crack.
So let me comment on that too, because I think it's important to look at the history of where we've been. It wasn't that long ago, 1970-ish, when there was nothing to do for a leukemia patient. Proof, just watch Love Story. Ally McGraw gets leukemia, there was nothing to be done for her. And now the majority of adult leukemics, and certainly the vast majority of pediatric leukemics, are cured. Now that happened in many ways because of empiricism. That happened trial and error. We still don't understand why ALL was so easy to cure. So I think we go from that era of imprecise oncology that has a few standout successes to the era of precision oncology, and now we're going back again, right? The immune system is something we're not going to understand until we study our patients better, and that brings us back to imprecision. Every other week, it seems, some other type of tumor is ceding to an immunologic assault, which is wonderful for patients, but it means we have to step back a little bit more. So I think our idea, and this is for all of us in the room, how we explain this to the public, we have to be careful that we're not overhyping the promise of precision medicine, but at the same time, that doesn't mean we don't believe in it. Eventually, patterns have a tendency to fall out. It's almost like we forget sometimes that oncology sprang out of infectious diseases, and we don't cure an infection, we cure a certain type of infection. We don't treat an infection, we treat tuberculosis or pneumococcal pneumonia or whatever. That level of precision in diagnostics and in therapeutics has to go together. And I actually think it's still a big challenge for all of us in the room to develop the data, phenomenologic, phenotypic, genomic, epigenomic, all of that we can put together to come up with these orthogonal approaches that will eventually give us the tools, I think, that will help more patients, cure more patients, and it really are very similar to what the panel before has said. We're in this for the long run. We have a few big hits of success, but we've always had a few big hits of success over the past 20, 30 years. I think we're going to see more of them, and I think they're going to be coming faster and faster. Jay, what do you think? You're in charge of one of the big armies fighting this fight, too.
Yeah, as I listen to this conversation, I think to myself, we have to beware of these paradigms because they lead to a paradigmatic way of thinking. And this, in some respects, can make you blind to the next breaking wave. There's no question that the development of Gleevec for chronic myeloid leukemia not only established the expectation of what science can do to deliver on the promise of cancer therapy, but it also elevated society's expectation of what we can do as drug innovators. What it led to, which was not all good, was the belief that with gene-directed therapies, that one by one, all of these cancers would fall. And it makes it feel as though immuno-oncology is this new breaking wave when it's been with us for decades. I'm a stem cell transplant physician. Donor lymphocyte infusion that works in many aggressive leukemias is just one type of activated T-cell directed therapy. So we need to learn and absorb these paradigms, find ways to combine these paradigmatic agents, but always skate to where the puck is going. Is it genetic guidance? Is it the hour rod on plain microscopy that tells you AML versus ALL? Is it a decision point of a phenotypic measurement of cell death proclivity, as Tony Latai has innovated at Dana-Farber, that guides small molecule selection? Is it some combination of all of these measurements that together create and establish the rationale for therapeutic development? This is where the puck is heading. We all talk about combinations, especially as regards immune agents, but it's always with some measurement, some mechanistic guidance, of which I think genotypic information is a powerful, maybe most powerful, but just one type of information.
Mm-hmm.
Jay, I want to stick with you for a minute and talk about novel mechanisms. So a lot of the discussions around this meeting has focused on novel mechanisms, and we've talked about a few. Are there new major novel mechanisms around the corner or areas that we should be thinking about putting in the armamentarium against cancer?
I'll take that in two directions. One is therapeutic mechanisms of action, and then another is paradigms of drug delivery. And maybe I'll just focus on the first in light of the conversation we just had around gene-directed therapy. We can't be too surprised that we haven't cured cancer when we don't as yet have therapeutic responses to the most common genetic alterations that cause cancer. We shouldn't be too surprised. And one of the luxuries that we have at NIBR is that with this longer horizon of discovery science and a real large group of talented, fully trained scientists, we can envision what technology needs to be in place in order to then innovate a distinct therapeutic response. And having come from academia, it's a very luxurious way of thinking to not have to worry series A, B, C, W, X, Y, and Z, or whether I can renew my R01 grant. Is it possible that we need to, as a field, set up the chessboard? Do we necessarily know what the cure for cancer will actually even look like therapeutically? And when you think about definitive medicines that have recently arisen, like engineered auto T cells for pediatric acute lymphoblastic leukemia, you immediately must consider that we may not as yet have the therapeutic technologies necessary to respond to the actionable events we already know, let alone those that are just beyond our reach of speculation. And so we spend a lot of time at the whiteboard trying to envision what new therapeutic science is required to deliver new therapeutic agents. You've heard surely about DNA-encoded libraries that dramatically expand chemical space of compound registries from millions into billions. We've innovated molecules that act like enzymes, recurrently degrading their protein target, though themselves not being degraded. There have been great innovations recently in genome editing that opened the door to engineering the human immune system in a way that was completely not fathomable several years ago. So as a field, if we can skate to where the puck is heading, and Bob, I know your group thinks a lot about this, if not exclusively about this, then the outlook is quite optimistic.
Great. Thanks, Jay. Let's talk a little bit about the clinic now and novel approaches to clinical trials. We all know that the cost of developing a new molecule is prohibitive by anybody's standards and rising. Our success rates are good, but could always be better. And obviously the time it takes to develop a new cancer drug, while in certain cases has shortened considerably, has not uniformly shortened. So how do we think about this? And maybe I'll start with George and then move to others. How do you think about new clinical trial designs? What excites you about interactions with regulators, if anything? And how do we think about that in the near term?
Well, I actually think that the FDA is in a wonderful position right now with Rob Califf. My experience with the FDA has been almost uniformly positive. They are looking to us, all of us, industry, academia, the public, to say, let us try to do this better. And actually, we are the envy of the world. Other countries in the world look at our FDA and say, yeah, you know, they're approving drugs, they're still focused on safety, they're still focused on old-fashioned things like efficacy. You know, we all want drugs that work. It does none of us any good if a drug doesn't work. So the answer there is, how can we best tell that to patients and make patients feel like participation in clinical trials really is something that is in their power, that we make it easy for them, that we get so many more patients onto clinical trials. You know, and that is, in many ways, a logistical challenge. That could be a moonshot issue. That could be a pure engineering issue. You know, at our cancer center, at the Dana-Farber Harvard Cancer Center here, Keith and I are working very hard to re-engineer the processes so that if you have a great idea for a clinical trial, it doesn't take 2 years to execute on it. You know, by that time, it's already stale bread on the shelf. So we are trying very hard to focus on the activities of our wonderful resources, not just here, but other great cancer centers across the country, as well as our community oncologists. Our colleagues in the community want to contribute to this. And so reaching out to them, making a certain type of program where trials are done in the community, other trials are done in academic centers, some can be shared, some of the more complex ones, certainly the CAR T cells right now are all in academic centers. I have no doubt that that's going to work, so we have to worry about really disseminating that kind of technology out and then focus on what we've done in academia for a long time. Focus on efficacy, but then make sure it's effective. Make sure that we're doing things that really matter in the real world as well. For me, it's all about having this technology. Just like Jay said, we might not even begin to imagine the kind of technology. You know, getting cells to be their own little 3D printers, getting them to make the kind of blocking agents inside with the kind of DNA origami that you can imagine transfecting into cells. There's all sorts of cool stuff that we can do. The clinical trials themselves aren't just about the simple version 1.0 of saying, okay, what's your genotype? Do you have the right genotype? You get this, the NCI match trial and other things. That's not a bad place to start, but it really is just the place we're starting. I think we're moving beyond that as fast as we possibly can. Keith, I don't want to take too long, or Gary?
No, I guess the only comment I would make is in terms of clinical trial design, I think we're increasingly trying to be creative about how we do that. I think FDA has been actually remarkably responsive to that. For me, one of the challenges is getting away from some of the established frontline therapies, many of which are cocktails of cytotoxics, and really having pancreatic cancer as a case in point of having to go into patients who have failed those frontline therapies and typically who are pretty beat up at that point with pretty poor status, and yet you know those first line agents are palliative at best. You're not going to cure anybody with them, and maybe finding more creative ways about getting patients on earlier lines of therapy with novel agents.
Yeah, I'll use this as an opportunity to shoe in a couple of points that I preach a lot about, is religion. It's, I'd say there's two major issues. One is that diagnostic technology and drug development need to co-evolve in a more fluid way. So it cannot be the case that we expect perfection out of diagnostics, but we accept 40% response rates out of therapeutics, right? We don't ever expect, perfection on that side, but for some reason, analytic validity needs to be just, spot on, absolute perfection, because it's seen as guiding, treatment decision making, and it's as though we're perfect in terms of the follow-on there. And I make that point to say that as we graduate towards NGS, multi-marker panels, which is, I would say, sufficiently in hand, at least the workflow to get there, that's probably not the hot button issue. But immune profiling certainly is still an inexact science, and yet one that needs to proceed, right? So just picking on that for a moment, when do we get to a world of a common PD-L1 platform? But beyond that, I mean, when do we start assaying inflammation of tumor at baseline and do that in a way that we think is not perfect, but reasonably good with 80%-ish predictive accuracy to say, okay, the real room for improvement is in this predicted non-responder population. Let's focus our attention there, and let's expect that we're not going to have complete diagnostic accuracy, but it's going to substantially enrich for being able to pick up hits in that population. Because just reproving that you can elicit responses in a patient population who are going to respond anyway to available therapy is obviously a trick that none of us are really after. So I think that's one piece. The other one is that everybody nods their head and says, yep, it's HIV, that's where we're going. It's 3 drugs, 4 drugs. We've got to figure out how to schedule and dose and do that in a safe and effective way. But we don't collide drugs until a very late point, right? We still live in a world in 2016 where the repertoire of approved drugs, in my mind, is completely unsatisfying in terms of pairing novel agents with those and expecting that that's going to actually be the biologically most robust synergistic strategy. So we need to credential drugs early on to find out they're actual drugs, meaning they have at least some pharmacokinetic prowess to them. But then beyond that, we have to allow them to get to their rational combination partners at a very early point, and we have to really lower the activation energy to get there. And this is really, really hard when drugs are coming from all different portions of the sector, large companies, small companies, and the same. And creating an ability for drugs to collide early, and I don't mean randomly, right? This is you do your 1,000 cancer cell line screen, and you find this really interesting signal where the partner other drug really doesn't have single agent activity in almost any context, and certainly not in the one where you've now found it, in ovarian cancer, wherever it is. We need to be able to then quickly, as George was saying, we have operational improvements to get there, but we have infrastructure, field-wide infrastructure issues to get to the point where 3 to 6 months later, you're treating your first cohort of 20 patients. And I emphasize the 20 patients to come back to the beginning of the question, which is how do we do this all efficiently? Like how do we actually improve probability of success, but also really massively improve efficiency? We need to be somewhat swinging for the fences, or at least trying to hit the gaps with doubles and not worrying about kind of running out the weak ground out and hoping we make it to first. So I think we have to accept the issue that we're looking for big effects in small numbers of samples, but I think we all like to think that if we're going to get there, HIV-like control of disease, combination and cocktails of therapies, that we're looking for big effect sizes. So I think we have a lot of work to do in terms of how we improve the kind of environment in which we allow drugs to find each other.
Keith reminded me of one thing as well that we can all do. We can all support an initiative that's right now on the table in Washington, which is to have a cancer center of excellence inside the FDA. It's really interesting how biologics went that way, diagnostics went that way, small molecule drugs went that way, and they don't necessarily all have any cancer relevance or any cancer expertise at the FDA. The FDA would like this, and I think it's good for research, it's good for the country, and frankly, after this will come a neuroscience center of excellence and some other very positive moves, because I can't imagine people at the FDA trying to review this field when they're in those little silos. Yeah. So one thing that doesn't cost a lot of money is just organizational excellence. And I think this makes really good sense, and I think it will happen.
Yeah.
George, I want to pick up on something you said before, because I think it'll be of interest to the audience if you could just expound a little bit on it, and that's the percentage of patients in this country that go on clinical trials. And my understanding, at least several years ago, it was at less than 5%. And I wonder, is that your experience, and how can we really think about that with all these new treatment modalities, all these new ideas about clinical trial design, yet we still have to go all around the world to sometimes find our patients. Maybe talk about that a little bit.
Yeah, well, I think most patients don't know enough about clinical trials. I think that's been a failure of medical care, to be honest with you. And I think as we wind up pushing more responsibility for medical care onto patients and their families. It behooves us as a society to really educate everybody about the fact that very often clinical trials are the best place to start therapy. And it will be the responsibility of the patient to ask their doctor, is a clinical trial right for me? And I'm not trying to make it sound too much like a TV ad of something in any way, but honestly, I do think that responsibility needs to be in the patient's hands because doctors are busy and they may not have the time to search. The frightening patient who comes in with a stack of paper this much, or here, take my iPhone, I Googled this and it says there are 150 different kinds of trials. Should I really go to the University of South Carolina for this? I think the answer is we need to do this better. Academics, industry, government, let's be honest, clinicaltrials.gov is very hard for patients to navigate. This is something that we have not done well, and I think we still need patients to trust their physician, but that means we need to give the physicians better tools to interact with their patients. I think that the numbers of patients that participate in really important clinical trials probably still is in the 4 to 5% range. I'm fortunate. I focus on a set of rare diseases. The population under care at Dana-Farber and Harvard tends to be a population that comes here to go on clinical trials. So in our Sarcoma Center, we think about 60% of patients are on various kinds of trials. Doesn't surprise me. That was true in pediatric oncology way back in the day. Frankly, even the rates of participation in pediatric oncology trials, now that we have standard curative therapies, those rates are dropping as well. So I think the responsibility is there not just to ask about it, not just to make it operationally easy for patients to participate and be sure that it's not a crushing cost burden for them to participate in those. It's also important for us, all of us up here, to design the best questions to ask. Build those into the trials so that as I'm talking to a patient about a study, I can say, this is a really important study, and I have equipoise about you participating in it, and I think it's fine. Just like Steve says, it's really important for doctors to be honest about what current therapies might offer and why we do research. We do research to make things better for the future. So that's a patient and family's way of contributing to what we're all in together.
Yeah, 2 quick points to that. So one issue, of course, is that we splinter the cancer population into smaller and smaller molecularly defined subsets, and not just by using one analyte, but some matrix of analytes. When we're in sort of straightforward phase 2 efficacy testing mode, we have to democratize the testing of therapies by actually going to where sparsely distributed patients live. So we have to leverage the network of sites that are interested and willing to participate in clinical trials, which are many community sites, many large practice sites who do plug into the cooperative group system, but not into industry so much, right? Industry goes to the recurring partners in their execution of clinical trials for very good reason. So I think there is a need. It's not just genetics. There's other measures by which you can think of this approach. Of really democratizing access to these therapies for very straightforward, plug-and-play, user-friendly types of phase 2 questions that can be tested that would enhance participation, not just in therapies that might or might not be relevant, but we think with really high likelihood are relevant to that population of patients. So there's ways of thinking about getting to that day, and NCI Match is a moment in the field in terms of trying to get to that as an example. The other point is that you shouldn't have to come to Boston to figure out what to do about your L576P mutation in GIST from George Demetri, or the acquired resistance mechanism that's come on the back of therapy. And George knows in about 3 seconds what he thinks the current repertoire of investigational agents are for the relevant resistance mutation that has cropped up following therapy aimed at that first mutation. You should have access to that information, and not just George. I mean, it would be George and 5 of his colleagues who would, quickly agree, yep, there's this one approved drug, but after that, there's these clinical trials that we think actually are featuring therapies that are really high priority. So getting access to that information so people could then navigate the 473 options that come up on clinicaltrials.gov and quickly zero in and say, okay, these are the trials that are most relevant. Either is or isn't possible for me to get to that clinical trial site and focus on that, but just demystify this whole issue about what does George think you should get for therapy, because it's not that challenging at any given moment.
And I appreciate Keith doing that shout out. The other thing is it's easier to find what's the best Chinese food in Worcester by asking Dr. Google than it is to find what's the best clinical trial for me. And I think this is where I think we can use some of the brains that sit in a different sector to help us do our job better, because it just hasn't been done still.
Yeah.
Sean, a question for you about pharmaceutical pipelines. You obviously run more than oncology at Amgen. And yet many of the new mechanisms that we're thinking about in oncology, whether it be immune mechanisms or epigenetics or metabolism and others, can apply broadly across a pharmaceutical pipeline. So how are you thinking about that with regard to your organization and teams of scientists?
Yeah, it's a great question, Bob. I think several years ago, I think it's now 3.5, 4 years ago, we actually made the decision to merge together our two biggest discovery research areas, which was inflammation, which is all the immune modulation therapies where we've had a very focused effort for a long time, and oncology. And we actually decided that there was so much promise in the immuno-oncology angle that we wanted to ensure that that was where the majority of the focus was in our activities. And we felt at the time that it would really shake things up to have someone who was trained as an immunologist and who worked on things like autoimmunity and infectious disease concepts actually be responsible for this area. Not everybody liked that idea, but we did it anyway, and I think it's been really great. And what you see is that it's incredibly energizing to scientists to be able to come into a group where they know that they could work sometimes at the same time in their labs on approaches to inflammatory disorders, autoimmune disorders, and cancer immunotherapy. And of course, you know, it's a yin and yang thing, and we find that there's tremendous synergy. As we all know, most of these checkpoint inhibitors that have been so commercially successful in oncology recently, they were in the pipelines of companies. Right. We had one in our pipeline we killed for autoimmunity, which was the exact polar opposite of what it ends up doing. But you know, this is very interesting area, I think, and we again see the cancer problem perhaps through a little bit different lens because of the breadth of work that we do, and see it as as more of an infectious disease kind of a problem with a debulking and mopping up kind of concept. So we've gone at it in a little bit, perhaps a little bit different angle. This led us to begin acquiring technologies like the micromet technology we acquired for bispecific T-cell engagers and the TVEC oncolytic virus technologies well before there was proof of concept in melanoma from ipilimumab. So, you know, when people were actually still scoffing largely at the idea of immunotherapy being commercially important for our industry. So we've achieved registrations for both of those products at this point. And I go back to something we talked about earlier. I didn't get a chance to comment. I think the FDA has been quite remarkable, as well as other regulators worldwide. I mean, you take both these examples, the first T-cell engaging therapy, our Blincyto therapy for ALL, CD19, and TVEC in melanoma. You know, these were small trials that were done at a time where the idea was highly speculative. They had surprisingly positive kind of results, but they were imperfect studies in many regards. I mean, the Blincyto studies are single-arm study, phase 2 data, you know, TVEC study had all kinds of issues with it, but the agencies saw their way to approving them, and I think that is our barrier at this point is much less about the regulatory environment than it was, let's say, 5 years ago, and much more about the kind of practical issues we've been talking about, like you can't get patients to, you know, 5% of patients enrolling in cancer clinical trials. I mean, that kind of problem is holding us back more than regulatory agencies.
Jay, I know you're still trying to learn the names of everybody at NIBR, but how are you thinking about the organization and the crosstalk among scientists?
Yeah. You know, not knowing a lot about NIBR before I got there, I might have thought that there would be siloing, that with major research centers in 7 of the most innovative parts of the world, that there would be imperfect communication. But I have to hand it to this Mark Fishman, who just retired as president of NIBR, and whom I'm succeeding. He is such a careful architect of science that as I've traveled each of these sites now over the last 4 months on the job, first 4 months on the job, I found the culture extremely familiar to what I was used to at Dana-Farber and the Broad Institute. Perfectly collaborative, very ambitious, really mission-oriented, and deeply interconnected. If there are silos at all, it's not among the people actually doing science. They recognize that they're in the midst of this intergalactic research infrastructure, and they find a way to connect to exactly that contributor within the organization who can advance their science. We look at the acknowledgement slides on these pipeline presentations. There's 40 scientists from 3 continents, and so I'm sure that it took a lot to build a type of culture and an institutional structure that would be so deeply interconnected, but I also think it's generational. The early career scientists today really crave connectivity, and they have an easy time finding it. Our IT systems are spectacular, maybe world leading, such that in my 7th day on the job, interested in a given target, within 5 minutes I can get the research presentation from the Shanghai site. And leveraging these modes of connectivity, we see, just like Sean, immuno-oncology medications rising up within Glenn Dranoff's IO shop, but also at GNF in San Diego, and also out of the ATI immunology site in Basel. I don't know if it's Novartis or if it's just the way that science is done now, but our scientists are, to me, it feels very familiar, very, very deeply interconnected. So what do you do with that then as a platform? You challenge the institution. And so I have already rolled out several initiatives that are NIBR-wide that defy the sort of typical organization by disease area or therapeutic area or content capacity, but provide immediate moments of inertia behind technology concepts that we think can catalyze important therapeutic science across the Institute and potentially around the world once broadcast. So there's a way, I think, to leverage the natural pace of human interaction right now, which is deeply open, deeply interconnected, to create, I think, a very competitive advantage in an otherwise what might have been historically siloed type of institution.
Great. I want to turn now to something that I think a lot about, and that's small companies and in niche areas of science. I'd be interested in hearing from George and Keith how your interactions have been interacting with small companies and helping them get off the ground. And then also from the pharmaceutical perspective, will you still be looking in the future to the small companies to help fill your pipeline? So maybe start with Keith and then George.
Yeah, well, I still see the small companies as doing a good bit of the beta testing and de-risking of totally novel therapeutic space, and there's a lot of that still to flesh out in terms of cancer biology categories. So with that, you get new therapy coming in an unvalidated arena in terms of the target, the pathway, the biology in its entirety. And so I think one of the major challenges is that it gets, if you start looking at the landscape of recent precedents and say, okay, well, this class of therapies established, this in the way of proof of mechanism, then they went on to proof of concept around single agent, and then moved on to combination in this way. You'd say, okay, well, there's in each of those categories multiple reasons why that doesn't pertain to you, right? So don't follow what turned out to be a third-in-class agent and the way at which it picked up the crumbs, from previous programs. You're first in class, and so you have new challenges, right? So you have to decide whether actually, taking this target engagement mechanism finding that your scientists have developed and really investing and making decisions around that is a way of decorating an academic paper, like sort of figure 5 in your JCO paper, or is it really go, no go? I mean, is it really critical? Because every unvalidated target has its unvalidated biomarker to go along with it, right? So you thought 80% inhibition was what you should be aiming for, it turns out you're off by, a log. So, in more time with more information. And so this issue about anguishing through, what are the levels of evidence that you can generate, with small companies, it is just oftentimes the case that although there might be some experience coming from different team members from other companies, other experiences, this is the first time they're coming together around this question. And so really trying to settle, these issues about what's critical for us to try to establish in the first couple steps of development, to me, that's the whole exercise. And part of the pleasure of being in this ecosystem, the Boston-Cambridge one writ large, is that you will always find a handful of people who have been focusing just on that target pathway in biology for their entire careers, have a ton of passion. And I, at least, am not smart enough to, predict and anticipate all of those issues in terms of where to set the bars, but I can certainly help be the dating service to connect them together. And I'm speaking to what it's like to operate in this environment, but I'd say in the field, that's what we face, right? It's bring the new concept, which has been living in a little bit of a quiet space in a silo, kind of until it's ready for the light of day, and really shine all the scientific light on it, which is to say, okay, what do we know about what points you can establish with this agent? Not just the window dressing parts. What are the really mission critical early development go, no go decisions? This is a learning enterprise on the academic side as it is for industry in the small company space.
My thoughts on this are that we're seeing a maturation of this whole field of cancer science research. And I actually think that after cancer science, hopefully, I think maybe we saw it first in cardiovascular science. Now it's in cancer science. Hopefully it'll move to neuroscience, where we're still groping around blindly in the best I can see. I guess my point here is only in cancer science that small companies still are very innovative. That they're small, they can take a risk. It's remarkably expensive. I think that's the other thing. So they can take little risks. I remember vividly when a little company on the West Coast called Plexicon, I was their scientific advisor. We were developing a BRAF inhibitor at the time. And ultimately, they got bought. And a company before them, Sugen, had gotten bought by Pfizer. I remember when Sugen got bought, and first by Pharmacia and then by Pfizer, I said, isn't this great? You guys have the brains, you have the innovation, they've got the money. And I thought that would work. And honestly, the cultures were different enough. So I've learned a lot from those naive days of mine, but I actually see a lot of experimentation going on in a company the size of Novartis, doing the kind of clever, creative things they're doing, the kinds of things that I saw Genentech do. Everybody wants to go back to the heyday when Genentech was, its own little thing, and it wasn't, G West, G Red, whatever. It wasn't G this, G that. So I think we're in a great time of that kind of social engineering as well, because everybody wants the thing that will work the best. And I don't think any of us know how to get there. I love the security of a big company that has money to actually do things, because a lot of academics, our trustees look at me and said, you've been part of a lot of FDA approvals. Can you do that with something we have? And I say, it's really hard. I could blow the whole endowment with a few bad bets. And I think that's the key. This is still a practical matter, not for the faint of heart. And it's risky, but thankfully our field has been blessed by a lot of people, many of you in the room maybe, being willing to invest in this for the good of patients, as well as hopefully some, doing well on the other side if things work.
How about from the pharma, Steve?
Yeah, I think obviously there's a lot more science going on, no matter how big you are outside of your walls than there are inside. I think to George's point, very often there's a level of creativity or innovation that pops up in a small startup company that simply wasn't on our radar or wasn't within our bandwidth. We pay a lot of attention to basically trying to stay connected with and mine that space, looking for some sort of transformative breakthrough, or perhaps just some chemical equity on a target that's of interest to us already. I think we spend a lot of time outward looking and are very, very open to bringing things in. One of the tensions I often see is that from the small company's perspective, they want to push their compounds as far as possible to create value. From our perspective, we'd rather engage as early as possible to make sure that you have a very, very good pharmaceutical product, which isn't just about great science. There's an engineering component to that. I do think there's a little bit of tension around that piece, but I certainly see the relationship between ourselves and multiple smaller companies as being very fruitful and geared up to be beneficial for both sides.
Great. So I think we're just about out of time, so let me maybe just close by saying I think you've heard from the panel, and I certainly feel this way, that the future is bright in terms of cancer drug discovery and development. Lots of innovative new ideas, team orientation, groups getting together, a regulatory environment which has been really dramatically favorable over the last several years and innovative. And we hope that everyone, first of all, I hope I'm right, and second of all, I hope everyone else shares that sentiment. Thank you very much.